Hello, welcome to my webinar for optimizing the extraction of human microbiome samples. My name is Dominic O'Neil. I'm the director for R&D. My responsibility is microbiome product development at QIAGEN. So just before we start with the presentation, I'd like to provide the quick legal disclaimer. I'll be talking about different QIAGEN products here, and the products that I show are intended for molecular biology applications and not intended for the diagnosis, prevention, or treatment of disease. Okay, and then to our agenda, I'll have a brief introduction on microbiome and why microbiome studies are interesting. I'm sure that you're all very well aware of this already. And then I will talk about optimal DNA extraction from stool samples with the QIAamp PowerFecal Pro kit, which we really do believe is the new gold standard for extracting DNA from stool samples. And then the next point is different sampling techniques and automation for the QIAamp PowerFecal Pro kit. And then the last part of the talk will wrap up with the application of this kit to other human microbiome samples outside the gut. So the microbiome is, of course, a fascinating subject of study, and new insights are being made consistently. One of the first major projects to investigate it was, of course, the Human Microbiome Project, which really showed the diversity of microbiomes that live with us and in us and on us, and provided the first insights into the depth of additional biological functions that these microbiomes provide in the second genome that we carry, and then also started to illuminate some of the health effects of it. And so, of course, it's fascinating to start looking at microbiome samples in order to understand what effect that has on humans. And of course, if you're doing a microbiome experiment, it's very different than simply looking for a single pathogen. So when you're looking for a microbiome sample, you pretty much have to do sequencing downstream, and you're mostly looking for answer one of two questions: which organisms are present, and what is their relative abundance? So if you're looking at the community that's present and which organisms may be providing a function within that community, is on the one side. Looking at DNA. If you're doing bacteria and archaea, you're probably interested in looking at 16S rDNA, or metagenomic will also provide you that information. You can get fungal information off of the ITS gene sequencing, viruses through metagenomic sequencing as well, and all of those are DNA-based analysis methods, and they will really tell you which organisms are present. You can get much more information out than you can with traditional culture methods and so on. On the other hand, if you're looking for functions of the community, you may want to look into metatranscriptomics if you're looking at RNA and messenger RNA that's present, or if you're looking at metabolites or protein characterization, you may want to be looking at metabolomics, proteomics. But for the purposes of the talk here, I'm really focusing on the left-hand side of this chart on which organisms are present, what's their relative abundance, and primarily around the 16S rRNA gene sequencing. And of course, like I said, mentioned at the beginning, the entire talk is around optimization of DNA extraction from human microbiome samples. So the questions are, what really matters when you're trying to do DNA isolation from microbiome analysis? And of course, the first point is that yield is critical. The more that you can get out of your sample, the more analysis that you can do, the higher chance you have of detecting low abundant species, and you can just simply be more confident in your results if you really know that you're extracting all the DNA that's present in your sample. You need to be aware of inhibitors. So especially in stool samples, inhibitors are a major challenge. There's lots and lots of inhibitory samples, which can then have a negative impact on your downstream analysis, particularly considering that most of the metagenomics or 16S analysis is all PCR-based or enzymatic reactions. And the inhibitors that are present in stool samples have a very dramatic effect on these downstream analysis, so your prep needs to be able to handle these. And then the last point is that you really need to lyse efficiently. The microbiome is made up of many highly diverse species with very diverse cell walls, and therefore also very different requirements for breaking them open. So depending on what you pick for your lysis, you can also bias your prep very strongly towards certain types of bacteria or fungus and miss other ones entirely. And we've found that that this is really a critical point that we've spent some time on optimizing. So in light of these 3 points, it's really what we've done in releasing the PowerFecal Pro, DNA, and DNeasy PowerSoil Pro kits. So these are second generation kits following on from the PowerFecal and PowerSoil kits, which have become very standardized for microbiome samples, originally from MoBio, and now QIAGEN has a second generation that we have upgraded in various ways. And the upgrades involve adding a new lysis tube. So there's a new lysis matrix that we have, along with a new lysis chemistry. And the inclusion of this really improves our yields dramatically, in some cases up to 20-fold. I'll show you some data on that. We've streamlined our inhibitor removal technology. As I mentioned, that's also highly critical. And the new IRT is faster, removes more inhibitors, providing a better downstream NGS PCR performance. And then the last point is, again, associated with these new PowerBead Pro tubes and the new lysis chemistry that we've implemented, is that we get a better lysis of the microbes in the sample, more uniform, and a higher detection of more species than previous. So we really do believe that these Pro kits provide a new gold standard and give you the best representation of stool and soil microbiomes. So as I mentioned, the inhibitor removal is a critical point, and that is shown here, the mechanism that we do it. So all of the QIAGEN kits which have power in their name somewhere, so the PowerSoil, the PowerFecal, the Power Microbiome, any of those kits include IRT. And IRT is a method of precipitating out inhibitors during the prep so that you don't have to do a post-elution cleanup, and separating the inhibitors very efficiently from the DNA so that they don't disturb you in your downstream technology. Now, down below, you can see an example of what happens when you do a sample prep with IRT on the left, with the clear eluate, and without IRT on the right, where you have a very discolored eluate. And the reason for this is that a lot of the inhibitors that are present in these kinds of samples are molecules that act very much like DNA or are very strongly associated with DNA. And that's, of course, also why they're so strongly inhibitory for your downstream PCR, is because either they'll bind to your enzyme or they'll bind to your DNA, preventing the enzyme from binding to it properly, and thereby significantly decrease your yield or your signal in the downstream PCR. And that's what's shown there on the right. The same sample with IRT, you're getting CTs around 10 to 12, and then once you take that same sample and run it without IRT, in one case, you're getting a little bit of signal around CT 15, 16 or so, and it goes up very slowly. And in the other, it's still up in the late 30s. So you can have really dramatic decreases in the performance of your PCR if you are not able to remove the inhibitors out of your sample. And that's what's done with these new kits very efficiently. But I also mentioned that we have dramatic increases in yield, and here's an example of that. So this is a test where we took 3 different donors, prepped 200 milligrams of stool sample using some commercially available prep kits, as well as the PowerFecal Pro kit. And you see for 2 of the samples, for donor A and donor B, you see dramatic increases in the yield that we get out. So that's where we get the up to 20-fold increased DNA yield. And we're getting, with the other kits on the market, we're getting very low yields for those kits, and then we get very stable yields for the new kit. And donor C, we do get an improvement still with the new kit, but it's not quite as dramatic as with donors A and B. And that's interesting to know a little bit about the background here, in that donor A and donor B are both stool samples from older individuals, and donor C is a stool sample from a younger individual. This is a test that we did to see whether or not we could find interesting differences between stool samples from old or young individuals. And what we found was that as a general tendency, now this is a limited sample size, and of course it's not a population study or anything, but we did find generally that stool from older individuals was more difficult to lyse, had a higher level of inhibitors, and saw a greater improvement with our new chemistry than stool from younger individuals, which generally tended to perform fairly well in almost most methods that we looked at. And that's what we see here as well. The dramatic increase comes from these difficult samples. And keeping that in mind, these are the same 3 samples, what we see is that when we compare it in the 16S sequencing, this is again doing it with the UCP PCR mix from QIAGEN and then a 16S sequencing over Illumina and analysis through CLC genomics. The two difficult samples, sample A and sample B, which were the stool samples from the older individuals, saw a marked increase in the diversity that we were able to capture with the Pro chemistry. So the blue lines up at the top, where we go from plateauing out at around maybe 300 species for sample A and around 200, 250 species for sample B. In both of the other cases, once we use our Pro chemistry, we're able to detect up to 500 different species. Now for the easy to lyse sample, sample C, there's very little difference between the two chemistries. So what we're seeing is that for some of these samples that are difficult to lyse, we really are able to break open more and more different bacteria, if they're present, than we are able to do with competitor or with legacy chemistries. And if you dive down into the data a little bit and look at it, in particular, what we're seeing is we're finding many more different species of bifidobacteria, as well as improved detection of archaea, in particular methanobacteria. And these are difficult to lyse samples, and so it was very exciting to see that we're able to find them at a much higher rate once we use this new chemistry. And I mentioned as well that you can do fungal sequencing, fungal PCR. This is here a fungal PCR look using at a 18S pan-fungal assay. And again, moving to our new chemistry, to the PowerFecal Pro chemistry compared to the legacy chemistry, we see a dramatic improvement in the extraction of fungal DNA. So with the legacy kit, we're getting a CT in the high 20s. And then with the Pro chemistry, we're getting a CT in the high teens. In both cases, it's the exact same sample being used, which means that the Pro chemistry is able to improve our detection by approximately 10 cycles compared to the legacy PowerFecal, thereby really dramatically improving sensitivity of fungal detection. Right, so now I've talked a lot about just the sheer yield and purity and IRT improvements that are possible through the Pro chemistry. But of course, there's other challenges as well for doing stool DNA extractions. And one of the major ones is handling. So as anybody who's done a lot of it knows that doing stool extraction, weighing out stool samples, whether they're thawed or whether you're chipping frozen samples and trying to move those things into small B tubes to do your lysis, it's a very tedious and messy and unpleasant process. And so in order to streamline this process, we wanted to investigate the effects of using inoculating loops to transfer sample rather than trying to weigh out each sample individually. So we looked at both 10 microliter and 1 microliter inoculating loops and transferred the same amount from the same stool sample, either weighing it out to get exactly 100 milligrams or simply taking a chip of frozen stool in the 10 microliter loop or the 1 microliter loop and transferring it into the prep. And what we saw was quite interesting. First, the yields were much more stable than we were expecting. We were expecting a higher variation on yield as we went to the loops rather than into the weighing method, but it looks to be a very stable method to do this. And the yields coming from the 10 microliter loop were surprisingly similar to weighing in 100 milligrams of sample. So for the 100 milligrams of sample, we're getting from 5 to 15 micrograms of DNA out, while for the 10 microliter inoculating loop, we're getting maybe 5 to 10. And for the 1 microliter loop, of course, it's lower, where we're seeing maybe 1 to 3, because we're transferring simply lower amounts of material. However, even with the 1 microliter inoculating loop, we're getting sufficient DNA out at the end that we can do good analysis. And we can see that on the next slide. We, of course, took the DNA that we extracted here, did a 16S PCR with the UCP multiplex PCR kit on the V4 region, sequenced it all on the MiSeq, and analyzed it in the Microbial Genomics Pro suite. And this is the OTU tables coming out of that test, where we did 8 individual biological replicates. So it's 8 individual weighings or transfers with the loop of the different donors, and did 8 individual extractions for each one, individual sequencing and libraries. So these are true biological replicates, and what's really noticeable is how consistent the method can be. So transferring with the loops is still a highly consistent way of finding the diversity in your sample, and whether you're using even a 1 microliter loop or weighing in 100 milligrams of material, the same information content comes out. We see that here in the OTU tables, and we also see it in the diversity plots. So if we do alpha diversity and beta diversity, we see no significant difference in the alpha diversity across the replicates, whether you're weighing in 100 milligrams or using the loops to transfer. And on the beta diversity plots, you see as well that the samples cluster very strongly based off of the donor and not at all based off of the method, which means that we're quite confident in recommending that the inoculating loop transfer method is a very simple and effective way to improve the sample transfer portion of the DNA extraction method. And we recommend that for people who are having trouble with the exact weighing out and the amount of effort that takes. So, but the improvements for your DNA extraction are, of course, not limited to sample transfer. The sample extraction itself can be automated, and we've done that in multiple different ways. We have different instruments for the sample homogenization, from the standard vortex adapters, which can do up to 24 samples, to the PowerLyzer 24, which can also do 24 samples in a faster method, to the TissueLyzer 2, which can do up to 96 samples, or even 192 if you do 296 well plates, which are also available as an option. And then to do the downstream analysis, we of course optimized the QIAamp PowerFecal Pro kit on the QIAcube, where it's the exact same kit that can just simply be automated on the classic Cube or the Cube Connect. But we also have separate kits available for the DNeasy PowerSoil Pro on the QIAcube HT, or the QIAsymphony PowerFecal Pro kit on the QIAsymphony. Both of these kits, the DNeasy PowerSoil Pro and QIAsymphony PowerFecal Pro, use the same upfront lysis IRT chemistry, and they've simply been adapted for optimizing the extraction on the instrument downstream. And these enable, of course, higher throughput and more parallel processing of samples if it's needed. And of course, it's only useful if you get the same good results out of the automated method that you do from the manual method, and that's shown here for the QIAsymphony. So we looked at the diversity and community in stool samples across 4 different donors in triplicate, either manually or on the QIAsymphony. You can see that in the OTU table. You can see very consistent profiles coming off of both methods, and a distinct profile coming from each individual. And that's, of course, mirrored in the beta diversity plot to the right, where you can see that the sample donor clusters very strongly, while the method seems not to make much difference. So there, we're also very pleased with the performance of the kit on the QIAsymphony, which provides an automated way of doing up to 96 samples in batches of 24. Or on the QIAcube HT, which is a 96 well plate-based method on a vacuum instrument. And there again, we see exactly the same results if we use a stool sample. The OTU table is on the right in this case, and the alpha diversity on the left. And the alpha diversity, you can't even see the 2 different charts, the 2 different colors, because they overlay each other almost precisely. And so we get, again, identical methods, whether we do here in 96 well format on an automated platform as we do if we do individual spin columns. So we're very pleased with the improvements that offers as well for the workflow. And then I come to the last part of the talk, where I'd like to mention a little bit about the use of these kits for samples other than stool. All of the performance data has been for stool, but still probably the most important sample type for looking at human microbiome, as the majority of the bacteria that live on us in us are carried in the gut. However, other sample types are, of course, interesting and provide also distinct microbiomes. And so we wanted to investigate whether or not we could use the QIAamp PowerFecal Pro kit for samples other than stool. In order to do this, we had an external service provider use our kits to extract from vaginal swabs, rectal swabs, skin swabs, buccal swabs, and saliva. All of these things were processed with the kits, and the extracted DNA was then amplified with the QIAseq 16S ITS panels, sequenced on Illumina MiSeq, and what I'll be showing is all the data from the B34 region, which we analyzed in Microbial Genomics Pro Suite on CLC. And we don't have any patient data or demographic data for these samples. So what we were really looking for was what came out of it. Was the sequencing successful? And if it was successful, was the information that came out of it biologically relevant? And that's the data that I will be showing here. So the first set of samples that I'm showing is saliva and buccal swabs. And here you can see pretty clearly that they're related to one another, as one would expect. One would expect they're both oral microbiome. The genuses, this is all shown, the OTU tables here are shown on the genus level. And we can see it's Streptococcus, Rhodia, Haemophilus, Neisseria, and Prevotella make up at least 50% of the community in both cases, in many cases up to 70, 75%, whether it's saliva or swabs. However, there's still some distinct differences that you can see. You can see that the swabs have generally higher levels of Streptococcus, while saliva has a higher level of Prevotella. And overall, it also looks like saliva simply picks up somewhat more bacteria species or genuses than the swabs do. However, they're clearly related. And what's also interesting compared to what we've generally seen with stool samples is that when we do oral microbiome samples, the oral microbiomes are much more consistent across individuals than, for example, something like a stool sample is. It's much more the same bacterial species, while in stool samples, we see much higher diversity within the samples. Now, the next set of samples was also interesting. These were vaginal swabs, and these showed a completely different pattern. They showed two very strong clusters. The first cluster was completely dominated by lactobacillus, and this is completely expected. That's the normal vaginal flora, and that's also considered generally the healthy vaginal flora should be dominated by lactobacillus in order to maintain the correct environment. And as long as that's also the case, it prevents infection with other species. And on the other hand, the second cluster of samples we had were nearly completely dominated by Gardnerella, which is a known pathogen implicated with bacterial vaginosis. And the Gardnerella, in this case, seems to have been strong infections, and so it nearly completely takes over the sample. We have one sample which is a split between Lactobacillus and Gardnerella, and that could be an infection that's either just starting or perhaps clearing. And then we have one more, which is also split between the two samples. Again, it's Lactobacillus at 50%, Gardnerella at 25%, but then the rest is highly diverse. And there's also some indications of this in the literature that when the normal flora is disrupted, opportunistic bacteria can come in and settle it. And the highly diverse bacterial community can also be an indication that something is not quite right in the sample. Now, skin swabs showed a distinct pattern as well, much like the vaginal swabs did. However, here we saw the two distinct patterns being that either the samples were dominated by Staphylococcus, which is of course a known skin flora type, or they were highly diverse. And it was not the case as in with the vaginal samples where it was dominated by a single pathogen. In this case, it's a very diverse sample that shows up in the skin if it's not dominated by the Staphylococcus. And as I said, I wish that we had either patient data or demographic data to say something about whether or not the diverse sample type here has an effect on health patterns. But it's certainly interesting to see this pattern simply that once the Staphylococcus is no longer the dominant species present, it looks like a very diverse set of skin flora arise. And then as expected, rectal swabs show us the highest diversity. We see, much like with stool samples, we see much more diversity between the individual samples. It's not as in with the oral microbiome where we see very much the same community members present across all sample types that we took with only minimal differences. here it's really highly diverse and highly individual from individual to individual. But as with everything else that we've looked at, the species or the genuses that we find here are exactly the ones that we would expect to see in a rectal swab. They're gut flora. There's some skin ones involved here as well. But again, I think this is also a very reasonable result for the rectal swabs. And then the last point here is the alpha diversity. It also showed a very interesting pattern where vaginal samples showed consistently a low diversity. That's exactly what we would expect after we look at the data and see that they're dominated by either the one or the other genus, except for there's, I believe, one vaginal sample that is a bit higher, and that's, of course, the one that had that highly diverse 25% representation. Skin sample is also generally a low diversity sample, right, with maybe 40, 50 different species detected, but with a couple skin samples which had a higher diversity. You can see those with a steeper curve, the light blue lines. Buccal swabs are the next most diverse sample, and that was also interesting to me to see that the buccal swabs in general also showed lower diversity than saliva. Saliva showed consistently a higher diversity, and of course the rectal swabs showed the highest diversity, and that's also completely expected. So we were very pleased with the consistency of the results and the fact that they all made biological sense. And the beta diversity also confirmed the consistency of the extraction. So here we're showing principal coordinate scatter plots. On the left-hand side, it's a two-dimensional one. The same data as a three-dimensional one is on the right-hand side. And that's just the limitations of showing a three-dimensional plot in a two-dimensional medium here. But on the two-dimensional plot, I think you can see quite neatly that the buccal swabs and saliva samples all cluster together, that they have a similar oral microbiome type, but there are distinct differences that one can see. Buccal swabs are more like buccal swabs and saliva is like buccal swabs, regardless of who the individual came from. Rectal swabs are spread out more, which is again what we expect to see, just simply based off of the diversity that we saw, while skin swabs make their own small cluster with some outliers, and vaginal swabs make two distinct clusters, whether it's a lactobacillus-dominated or Gardnerella-dominated sample. So again, we're pleased with the consistency of the microbiome extraction for all of these different sample types, and can recommend the use of the QIAamp PowerFecal Pro kit for extraction of all different kinds of human microbiome samples without reservation. Right, and so we'll end on this note that QIAGEN really strives to provide a complete sample to insight solution for unlocking your microbiome studies, from sample disruption, sample preparation, all the way through the library preparation, NGS, and data analysis. We strive to present you with the best possible methods to get the most information out of your microbiome samples. And I'd like to thank you for your attention and would take any questions. Thank you.